Chapter 11, Topic 1, The Human Host. In this recorded lecture, we're going to cover what is disease, resident biota, how we get biota, and where we don't have biota. The learning objectives for this topic are be able to differentiate between colonization, infection, and disease, be able to enumerate the sites where normal biota is found in humans, be able to discuss how the Human Microbiome Project will change our understanding of normal biota, and be able to point out how microbial antagonism can be helpful to the human host. Before we start on the microbiome, let's first define a few terms. The first of these is disease. Any disease is defined as a deviation from health. Note that this could be a genetic disease, an infectious disease, or even aging. A disease that is caused by a microbe is known as infectious disease. One last term to discuss is colonization. This can be a little confusing because we would assume that any pathogen would colonize within the host, but this is not entirely true. If you think about how an infection progresses, it is not through colonization, but through contamination. Generally, microbes of the microbiome are the ones that actually colonize the host. We have talked about the microbiome many times in this term, but let's review a couple of key points about it. First, the microbiome is a mixture of all three types of microbes we have talked about this term, and we honestly don't know the entire catalog yet. We are working on identifying it, and the more we learn about this symbiotic relationship, the more we come to appreciate how helpful these microbes are. While we don't fully understand the importance of the microbiome, we have been aware of it for a while. This has led to a variety of names including normal flora, biota, and likely microbiome. It is important to appreciate just how specialized these microbes are. They had to be able to colonize in one of the most diverse environments we offer as hosts, and be able to replicate and survive. Not only this, but given what we will learn about the immune system, these microbes have to evade the immune system's detection methods. Due to both these features, these microbes have evolved right alongside us, creating a very unique relationship between us and our microbiome. So how do you and I acquire our microbiome? We start establishing our microbiome at birth and shortly thereafter. This establishment can come from the birth process or through breastfeeding. Even being held by others early in our lives helps establish this microbiome. This microbiome can be fairly stable throughout our lives, but can be altered by drugs or by other environmental factors. For the most part, any surface area that has external exposure is likely to be colonized by microbes and help establish a microbiome for that area. This includes parts of our body that we normally think of inside. For example, your digestive system has contact with the external environment, and it has a very diverse microbiome. However, not all surfaces of our body are susceptible to colonization. These tend to be internal organs and fluids. This is why it's so important that we de-germ surgeries or any kind of puncture wound, as any microbe can lead to an infection even by normal microbiota. So let's go over again the learning objectives for this topic. Be able to differentiate between colonization, infection, and disease. Be able to enumerate the sites where normal biota is found in humans. Be able to discuss how the Human Microbiome Project will change our understanding of normal biota. And be able to point out how microbial antagonism can be helpful to the human host.